Now, biotech company Imagion Biosystems has recently announced an update on the progress of their first in-human HER2 breast cancer clinical trial. Interim data on their first 5 patients dosed shows that its imaging agent is safe, well tolerated, and is reaching patients' lymph nodes. Bob Proulx, Imagion Biosystems CEO, joins me now to tell us more. Bob, thank you for joining me. How are you going today?
Very well, thanks. Nice to be there.
Of course. Now, what exactly are you aiming to achieve with this study, and what will it mean for the industry and breast cancer patients if your study is successful?
Yeah, so today, when we diagnose patients for breast cancer, the first thing we want to know is it's spread to the lymph nodes. And unfortunately, the standard of care historically has been that we need to do a surgical excision or a biopsy of some kind to determine that. So we've been working on a non-invasive way to potentially demonstrate or detect whether or not patients have nodal involvement in their lymph nodes associated with their breast cancer. And so this phase 1 study that we're doing is intended to be that first proof of principle that our imaging agent could potentially be a non-invasive way to determine whether a woman has invasive breast cancer associated with the primary tumor. And so this phase 1 study, first goal is, of course, to demonstrate that, in fact, our imaging agent can be administered to the patients. It's safe, well tolerated, and then have enough data to support whether or not we think we should go forward with this in a larger round of pivotal studies to be proven for its use as a commercial imaging agent.
Of course. Now, I understand that you are still undergoing human clinical trials. So what is this process like, and whereabouts are you guys on in the process at the moment?
Yeah, so we currently have 4 sites in Australia that are helping to recruit patients into this study. As we reported in our interim results, we've been through the first 5 patients that we have full data on. And at this point in time, we're hoping to continue to recruit up to somewhere between 10 and 15 patients is our goal. At that point in time, we think that we'll have enough data to indicate whether or not it merits continuing to proceed into larger studies that would then be used for regulatory submission, or whether we need to go back and do some additional work, if you will, to optimize. So we're hoping to continue this year, by the end of this year, be able to have gotten through around 10 to 15 patients and be in a position to know whether we can continue to move forward with this.
Definitely. And how about the future? What does the future hold for not only the company, but the research that you're conducting? I mean, it's pretty groundbreaking, being a non-invasive procedure.
Yes, so this study in particular is the first time, as far as we know, that a magnetic nanoparticle has been used with a targeting ligand of some kind to non-invasively target specific types of tumor cells for the purposes of being able to do diagnostic imaging. And so we're very pleased with having gotten into the clinic, now starting to get actual human data that's giving us an indication as to how we might want to move forward with this first product for HER2 breast cancer. But behind that is the work that we're doing to now take that platform of magnetic particles and be able to put different targeting agents on, perhaps, for example, to target prostate cancer for men, ovarian cancer, and other types of solid tumors. And so what we're looking to try to do this year is, while we're gathering the remaining information from these patients we're studying in Australia, is to build that pipeline of other imaging agents where we could apply the same non-invasive approach to other forms of cancer.
Absolutely. I mean, it's very interesting. But let's just talk about the development of this new technology. What exactly is going on behind the scenes? How is this all working, and how is it changing the game?
So, one of the things that we've had up until now is imaging technologies in general. So we got 5 ways to image the body: MR, CT, PET, ultrasound, and X-ray. And they all do a very good job of being able to look at the anatomy and various, giving you very good resolution of various types of tissue. But historically, they've suffered from the idea that they can tell you that there's a suspicious area, but they can't tell you, for example, whether what they see is actually a form of cancer. We're all familiar with women having a mammogram. You see a spot, but is it a malignant form of cancer, or is it a fibrotic tumor of some kind? So what we're trying to do, what's behind the scenes here, is we're trying to then see if we can move basic imaging into what would be more normally thought of as molecular imaging, so that when we see a spot, we're actually able to say, no, in fact, we know that that's a particular form of cancer because we've got a targeting molecule on our nanoparticle that's highly specific for that type of cancer. So we're trying to transform imaging from being not just really good pictures in images, but actually being able to be used as a diagnostic tool, because we can confirm that the presence of our imaging agent is there because of a particular form of cancer.
Right. Well, Bob, thank you so much for sharing this update with us, and I can't wait to see what happens with the rest of the study. Thanks for joining us.
Great. Thanks for letting us share our story.